Hello, everyone. I'm Vince Calhoun, and I have a great pleasure to introduce Gustavo Deco for the next plenary. He is currently the director of the Center for Brain and Cognition and also leads the theoretical and computational group at the University of Pompeu Fabra in Barcelona, Spain. He actually has, in 1987, he got his PhD in physics. And so I thought I would start out with a joke about a biologist, a chemist, and a physicist who took a walk through the country, and they saw a cow, and for some reason they didn't know what it was. And so the biologist thinks for a second. He says, "I know what that is. That's Bos bovine." The chemist looks for a second and says, "It's just a carbon-based life form, approximately 75% water and 25% trace elements." The physicist stares blankly for a second and then says, "Well, I guess we could approximate it as a sphere." So now that does not do justice to Gustavo at all. His research is really trying to get at mechanisms, computational principles, basically trying to understand higher brain function in the context of both healthy and diseased brains. And he actually complements his PhD in physics with two other PhDs in psychology and computer science. And so I think you're going to be pleased with his talk. He's going to be talking about causality and brain dynamics. And without further ado, Gustavo Deco. Thank you.
Yes, thank you, Vince, for the nice introduction, and thank you, the organizer, for inviting me. It's really a huge pleasure, an honor to be here, and thank you for the opportunity to present here part of my research. So I will follow the preliminaries of Vince, and I decided actually a very risky thing. I want to speak about something which is relatively new. It's not published at all. It's also not finished. It's about how to promote brain transitions, and I will follow the physics approach. So I will try to approach a very difficult problems, like to promote brain transitions, assuming that I can approach this with a sphere. So let's see how we do that. So the plan of my talk is really, as I say, the main motivation is really to try to learn or to try to start to understand how we can promote transitions between different brain states in a stimulus-driven basis. And in order to do that, my claim will be that we need to know the underlying generative, causative mechanisms generating the brain dynamics. So, as you know, I mean, that was the business of whole brain models, and we started doing whole brain models in order to understand those underlying mechanisms generating the brain dynamics per se, just from a basic neuroscience point of view, also for clinical application in order to detect better biomarkers. But now the main focus is because we are dealing with a complex system and we want to manipulate that particular complex system, we need to understand those mechanisms or to extract those mechanisms in order to try to manipulate them. So in the first part of my talk, therefore, I will very briefly review the whole brain models and in particular present a new version of a brain model which are unifying many different perspectives, and then I will use that brain model in order to characterize brain state. So when I speak about the stimulus-driven transition, what I have in mind or my expectation is to use external artificial stimulation like non-invasive, like in the case of TMS, or invasive, like in the case of TBS, that we know that they affect the brain dynamics, but they are relatively simple. They are very, most of them, I mean, they are very local, and the idea is really how with this simple and local stimulation, we can really provoke transition at the global level of the brain dynamics. Just to motivate you, you know this type of videos, and you know the effect of TBS. This is a patient of Tipo Assis in Oxford. She has a dystonia, so you need how catastrophic behavioral at the motor level this type of disease. And she got implanted DBS, And you will see soon in a couple of seconds how she reacts after a couple of months having this DBS. So as you see, now she can move, and then suddenly, 18 months after the implantation of the DBS, she behaved cognitively and from the motor point of view absolutely normal. So just my last cartoon for motivation, because the idea is to promote this stimulus-driven transition between different brain states in order to use this also in a clinical context. Let's imagine that we have a healthy brain with a healthy brain dynamics, and imagine that we can describe this complex dynamics with some particular whole brain approach. And in this case, I'm using this landscape cartoon trying to symbolize that if we have some particular attractor characterizing the healthy working point of this complex dynamical system, then if we suffer some disease because of many different reasons, because we have alteration of the underlying structure, of the underlying system of neuromodulators, neurotransmitters, and then we have distorted this landscape, and therefore, instead of working in the right point, we are working in a disease working point. And the idea to use stimulation, or the first idea, the first intuition would be to design this stimulation in order to move the working point artificially, to move the dynamical trajectories artificially in order to try to recover the normal healthy trajectories. But our approach actually will be a little bit different, because starting from that particular distorted disease dynamical system, we will design the external stimulation in order to recover actually the form of the landscape, the characteristic of the whole brain dynamics, in such a way that the system naturally will then go to the normal working point, and hoping that homeostatically the plastic change that are promoted by this change of the landscape will recover the system. So far the motivations. Let's start now with the real business. So let's start speaking about a causal mechanism in whole brain modeling. As you know, the general strategy of whole brain models during the last 10 years, followed by many different groups around the world, were somehow to link through a dynamical model the underlying anatomy, which we are quantitatively specifying in terms of DTI tractography or similar techniques, and describing at the level of a structural connectivity matrix the different existing connections, the wiring underlying those coupling between the different local dynamical systems. Then you put your favorite type of local description of the dynamics, and we hope that this generate or simulate a functional dynamics that we can compare with the reality. In this case, for example, just using Bolt, but of course, and functional connectivity, but of course we are not restricted to this technology. We can use imaging, EEG, or even neurophysiological recordings in animals. And the idea is to explain how this link between structural and functional connectivity works. As you know, there are three main ingredients, as I say: the structure, that at the moment we will not touch, we will just assume as given. And then the other two complementary aspects are how to describe the local dynamics. And here we have really a variety of different possible choices. So describing really the dynamics at the very realistic biophysical level, at the level of spiking neurons with the realistic synaptic dynamics, or different type of reductions like neural mass models, or simply perhaps noise. And the third element is how to characterize that brain dynamics. At the moment, in the last cartoon, I just insinuated that we are assuming, and that is what in fact happens during the last 10 years, that these dynamics can be somehow resumed in this very particular measure that we call nowadays the static or grand average functional connectivity. So we are considering just a spatial correlation. But of course, there are other possible measures that could be more informative. So the main thing is that we will focus our attention in this first part in these two main questions: which is the local dynamics, which is more appropriate, and how to characterize the brain dynamics. Here I have, in a practically criminal way, tried to resume all the modeling works of the last 10 years. I divided this taxonomically in 3 different scenarios. The first one, where we just assume that the dynamic is extremely simple. It's a noise, so meaning that we have autoregressive models like the ones proposed by Messier, or linearization of mean field models, which are also mainly working with an assumption of the dynamics being just noise. Of course, we can refine the local dynamics and assume spiking networks or more sophisticated neural mass or mean field models. And the third scenario, so which all have the characteristic that they are working in the asynchronous regime, or models which are assuming that the local dynamic is oscillatory dynamics. The good news is that all models works, so meaning all models can explain relatively well the grand average static functional connectivity. The bad news is that all model works, so meaning that we cannot distinguish between them. So in fact, there were 2 papers of Messier recently showing that all these different models that I was describing before, in somehow they are more or less equivalent in terms that they can describe this link between a structure and the functional connectivity dynamics. So that is not astonishing, because if we remember this seminal work of Marcus Reichel and Mauricio Corbetta lab with monkeys, fMRI, and they were already showing that the functional patterns, the spatial functional patterns observed during resting state, in this case was light anesthetized monkeys, and the patterns of wires, the anatomical patterns, so the structural connectivity, is very similar. So meaning that for explaining the functional connectivity, just whatever you use is able to generate a good explanation of the grand average functional connectivity. In other words, does not mean that your models are simple, like noise. That means that your measure, the one that you are using, is not informative enough. And this is not astonishing, because as we know, there are many papers that just checking the relationship between functional connectivity and structural connectivity under different brain states situation. So when you use anesthesia in monkeys or sleep in humans, the correlations between these 2 matrices change. And of course, the anatomy is not changing. So meaning that this strange, strong relationship between the SC and FC that we observe in the models is not accurate enough. So as we know, and that's come from the lab of Vince Calhoun, some time ago in the seminal paper of Allen et al. From Vince's lab, which was proposed that, of course, we can try to take into account the time information in the form of the functional connectivity dynamics. So meaning that if we, instead of calculating the spatial correlation in the long window, we just assume a relatively short window of 30 seconds or 1 minute, and we slide the window, we see those patterns of functional connectivity, and we see how those patterns of functional connectivity change in time. The idea is now to detect, how we can unify these 2 different models, and if the functional connectivity dynamics is good enough for constraining in a better way, more than the functional connectivity, the real dynamics. So in this case, what we use is a very simple model which you try to unify the different scenarios, in particular the asynchronous noisy scenario and the oscillatory scenarios. We use a Hopf model, which is a mathematical form which is able to describe at the phenomenological level these 2 scenarios. So if we are on one part of the local bifurcation parameters, we have just noise. If we are on the other extreme, so very positive, we are in the oscillatory scenarios. If we are in between, we have a mixture between these 2 different scenarios. And it's turned out that by manipulating, the 2 parameters which regulate the models, namely the level of coupling between the different nodes and the position of that bifurcation parameter. Remember that if this bifurcation parameter is very negative, we have noise. If this is positive, we have oscillations. We try to fit the functional connectivity, the static one, and we see that there is a very particular region, a hotspot that have a good fitting, and we try to fit the functional connectivity dynamics, so this fluctuation in time, and the region is also very specific and consistent with the functional connectivity. Of course, you can try to fit other spatiotemporal measures like metastability, but let's us to concentrate only on these two. And what we see that in this particular region where we have the local bifurcation parameter at the edge of the bifurcation, where we have a perfect mixture between noise and oscillation, we have the best explanation of the static functional connectivity that we get anyway in all the different scenarios as we observed it before. But now we have a very informative measure in form of this functional connectivity dynamics, meaning that it's extremely informative for constraining the model. Of course, just very rapid, we can do a better job and determine really not only a general bifurcation parameter, but a local bifurcation parameter for each brain region using machine learning techniques that I don't want to go into the details now. I just want to show an application in the context of stimulus-driven modification of the global dynamical state. These are data that we obtained from UCL, where they have Parkinson patients with a DBS implantation in the subthalamic nucleus, and we have resting state with the DBS on and with the DBS off. And what you see here is just a distribution of those local bifurcations parameters. So you have to think that as a kind of generative parameters, which is explaining in which way the dynamics at the spatio-temporal level, because we are describing the dynamic with the functional connectivity dynamics, emerge. And this is the group of healthy patients, and as you see, most of the parameters are around 0, which is not astonishing, it's what we found before. So they are in this mixed region of noise and oscillations. When the DBS off, so in the pure Parkinson case, without a stimulation, you see how much distorted are the distribution of those generative parameters. So meaning that it's very informative, and meaning that it's really not just a local problem of the subcortical substantia nigra basal ganglia system. These are reflecting just the cortex parameters. So meaning that the cortex is strongly affected, and funny enough, and the DBS was not designed for that, because it was designed just to really compensate the problems in the basal ganglion system, so mainly the motor problems. What we see is that when the DBS is on, all those parameters are going in the right direction, meaning that they are trying to recover the healthy state. Of course, we can be, and I just put this here in order to give you a flavor of the type of manipulation that we can do with this model. When we have a relatively acceptable explanation of in which way the dynamic is generated, then we can also try to explain how this dynamic at the global level will change when we apply very particular local stimulation. In this case, if you perturb area by area, we can see how the different levels of perturbation could affect the whole brain, and to use that in order to design where is the best way of possible target for our stimulation. The next step is really that we can do even better. So instead of using just the functional connectivity dynamics, we can use LEIDA. That was a method proposed by Joanna Cabral. She just gave a talk in one of the symposiums. Where we decide to describe the spatio-temporal dynamics at the probabilistic state space level. The idea is very simple. You just take, like in the functional connectivity dynamics idea, and a snapshot of the dynamic. In this case, we just use a phase description. So we use a matrix containing the difference between the phases for each pair. So meaning it's a snapshot of the dynamics, and then we do this for all the different time points. And then the idea is to clusterize that. In the particular case of LEIDA, instead of clusterizing just the main matrix, we clusterize the leading vector of each matrix, and we can extract the different micro brain states, if you want. So our microstates, as we call even metastable microstates, because they are not stable forever, but as you see, they are switching, and we can calculate the probability of transition or even the probability state space, and we can use this description at the probability state level in order to adjust the model. It's turned out that we can do that, and the result is even more accurate, given that the probability state description is a better description of the global dynamical state. The working point is different from the one indicated by the functional connectivity dynamics, meaning that we will use that particular point in order to describe the states. The key issue here, is, and this is a proof of concept we will do now, or we will use this probability transition description for describing two particular different brain states. The first brain state corresponds to a resting state, a wake state in healthy humans, and the second to deep sleep, the N3 state. For that, we use the data from Helmut Slauf from Germany and Enzo Tagliasucci. Here we do a relatively small modification because we need to be a little bit more picky in the description of those states. Instead of using just one global coupling parameter, as we were using until now, we use an effective connectivity description. So meaning instead of assuming that the fibers effectively are all scaled with the same parameter, we assume that there is an independent description for that particular fibers, and we can put that in the form of a matrix, meaning that we can describe that by the level of effectivity of each existing anatomical connections. If we do that for these two states, we do a good job. This corresponds to a wake, this corresponds to a sleep, and we can have a good explanation of the functional connectivity dynamics, a relatively good explanation of the functional connectivity, of the static functional connectivity. But what is more important here, that we have a good description for each different state, and it's a different description at the probability state level. That we can describe just by the different state, and in this case we have chosen on purpose just a relatively small number of states, 3. Of course, you can try this systematically for a different number of states, and we have for each particular brain state, awake or deep sleep, a particular profile or probability description corresponding to that global state. Of course, you can describe that also at the probability transition level. So now that we have a particular effective model for this state and a particular model for this state, and each one of these models is describing appropriately the empirically observed probability state space, now the key question, and is the most interesting part of my talk, so this is the most important slide, I would say, is to study if in the model, at least in silico, we can start from one state. In this case, we start from the sleep state, the model that is able to characterize the probability state space empirically under that particular condition, resting, awake. And now the key idea is that if we can promote a transition to another state, in this case to the wake state. This is the reason why we call this awakening, this proof of concept. But of course the idea is more general. We can go from any state to another state, even from a healthy state to a disease state. That is the hope. Of course, those states are now characterized by a different profile of the probability state space. And now we can study systematically and exhaustively things that we cannot do empirically because of ethical reasons, because of statistical power, and because of time. But in the model, we can do this exhaustively and so accurate as possible. We can study different perturbations, different externally driven stimulation, and see if one of those stimulations with one particular strength really goes to the appropriate brain state so that we promote that transition. In order to do that, we use really the Hoff model that I presented in order to show you that this model is able to unify, to syncretize in a very simple model the different scenario, the noisy and the oscillatory scenario, but also this model is very simple and suitable for implementing this particular stimulus-driven perturbation for changing the global dynamics. The idea here is that when you decide to perturb one particular area, we do this actually in an homotopic way, symmetric way in both hemispheres, but you have absolutely freedom to do whatever you want. But when we decide to perturbate one particular brain area, then artificially we move this local bifurcation parameter in one direction or the other direction. So we have the parameter that we fit it empirically, let's say around 0, and then we can force for a while this parameter to be on the noisy regime, so meaning that we are imposing a destruction of the global dynamics, or we can move artificially this parameter to the right part of the dynamic, meaning that we impose artificially, again, in a stimulus-driven way, oscillations, meaning that we will promote probably the synchronization of the global dynamics. What you see here is the main result of this proof of concept is for different strengths of the applied perturbation, meaning different level of moving this local parameter, very little or a lot, very little or a lot in one or the other direction. In this case, we are using the synchronization scenario, so meaning that we are moving the parameter externally to the right, so imposing oscillation. And this is where we apply the oscillation for the particular parcellation. Here we use the traditional AAL. So you see here the 19 cortical and subcortical regions. And we are applying systematically this perturbation and see what happens. And to see what happens, we just here in the color scales reflect how far we are after applying the perturbation from the target profile at the probability state space. And for that, we use a very simple measure, which is a symmetrized version of the Kolmogorov, of the Kullback-Leibler entropy, which is a measure for distinguishing how far away we are from the target probability state space. So as you see, when we have here bluish, means that this probability is really well fitted, meaning that starting from the sleep model, by perturbation, you can promote in a pretty unspecific way, which is also somehow expected, because whatever areas you stimulate with synchronizations promote this awakening effect. Of course, not all areas are exactly so effective. Here some couple of areas which are not so effective. But in general, in a pretty non-specific way, for a given strength, you promote this transition to the right state. As I said, of course, we can use other protocols and we can use other transitions. That is what I was showing you before in the last slide. But now we go the other way around, from awake to sleep, meaning we depart with a model which fits empirical dates according to awake, so the probability state space, empirical probability state space corresponding to awake, and then we start to stimulate systematically, area by area, with different strengths, and we see how good we approach now the sleep state. In order to do that, we need to use another protocol, in this case the noisy protocol, meaning that we are moving now the bifurcation parameters to the other direction, so meaning that instead of promoting synchronization with the perturbation, we impose more noise in the system, so intuitively we can think that we are destroying the general level of oscillations. Of course, not all possible ways of perturbing the system works, and here I am showing two transitions that are not working. For example, if you try to go to sleep, from sleep to awake in a noisy way, so by promoting noise, you are not doing good. You are only spoiling the system. or if you do the other way around, trying to go from awake to sleep, imposing synchronization, you also are not able to do that. You are, again, spoiling the system. And that is somehow a posteriori, at least, also intuitive and expected. Of course, this gives us also the freedom to study, at least in silico offline, with the model, different level or different type of perturbation, not only in the style of the strategy and on which nodes we stimulate, as we have been doing until now, so exhaustive exploration of single stimulation of all possible nodes. But here we test the idea of applying multi-site stimulation. And this is again the case describing the transition from sleep to awake. Now you see the numbers instead of the color scale. And what you see is really by different stimulation of more than one site simultaneously. The advantage is that the stimulation is much weaker. So we use a relatively very weak stimulation, and we can provoke this transition really from the sleep state to the awake state by perturbing, in this case, 4 or 5 simultaneously different brain areas. And even we can understand why this transition was provoked, as we could have been done also in the other case, because we can study also at the level of the probabilistic state space on those metastable microstates, how the difference perturbation, in this case the multi-perturbation, is affecting the single probability state space, so that they are promoting really the transition or the approach of the whole probability state space from one case to the other case. And here what you see is there is mainly one state, which is the second state, that is really changing and approaching the relevant global description of the probability state space. So just to finish, a couple of words about the future. Of course, we were until now, and I think that was the strength and the take-home message of my talk and of the last 10 years of whole brain modeling around the world in the different labs, is we know nowadays, in a relatively simple way, how to describe this complex global dynamics in different brain states, not only resting state, not only sleep, not only anesthesia, not only healthy, but also disease, and also not only in humans, but also in monkeys. And the main idea was to unify that through the binding between dynamics, and we have here very clear take-home message, that dynamics should be flexible enough in order to incorporate in a flexible way that we can adjust afterward with experiments, the global parameters or the local parameters or the scenarios, if it is noisy, asynchronous, or oscillatory, with the underlying anatomy. But now there is a missing information. And that missing information, actually, if you were attending properly, was actually replaced by the use of the effective connectivity matrix. So meaning, in order to be able to describe accurately empirical data associated with different possible states, we need to use not only the simple global parameters and the simple local bifurcation parameter, but on top of that, we need to change the level of effectivity at the fiber level for each pair. And of course, we can imagine that those change in the effectivity of each fibers is associated, of course, not with the change in the anatomy, it's not with the change in the number of fibers, but with the change at the biophysical level and probably modulated by neurotransmitters. So the challenge for the future is to do these causative models in order to extract information about the generative mechanism for understanding the generation of the global brain dynamics and for being able to manipulate that and provoke even a stimulus-driven brain transition, but now incorporating more details, and this more details implies the system of neurotransmitter. This is, again, just another proof of concept. So this is what I was saying. In general, what we were modeling until now was the linking between these two systems. So at the empirical level, between DTI tractography, between anatomy, and between functional connectivity. Of course, now in a much more sophisticated way. It's not just static functional connectivity, it's functional connectivity dynamics, probability state description, and so on. But the model were basically unifying these two levels. Yesterday, Gitte Moser-Knudsen presented in her talk the results coming from PET studies, where we can nowadays, of course, have information about the distribution of the receptors binding. In particular, for example, in this case, for this proof of concept, we were using serotonin receptors, in particular the 5-HT2A, which is mainly stimulated by a particular condition, which is by using psychedelics, that I will show in the last slides. And we can incorporate this information in the model in the form of just modifying simply the gain modulation of your favorite neural mass model that you can use in the context of this linkage between structure and function, or even explicitly as a separate dynamical system regulating the dynamics of the neuromodulators. If you do that, I mean, you can do a better job, and that is my last slide. In this case, we are modeling the effect of LSD, a psychedelic, as you know, in the context of resting state. That's our data from the Imperial College coming from Robin Carhart-Harris. And the idea is that if you manipulate those neural gaining parameters, those parameters which are associated with the neurotransmitter system, this is the blue curve. This is just the fitting at the functional connectivity dynamical level. So you have a better fitting. You are doing a good job. Much better than if you ignore the neurotransmitter system. And of course, much better if you excite those particular receptors that you are trying to incorporate in your model, so namely the serotonin 2A receptors. If you are using just placebo, this is the green curve, you are doing nothing. Even more, we are showing here by just reshuffling the data of Gitte that really the distribution of the receptors is absolutely fundamental, because if you reshuffle those information and you insert that in the model, you are not doing such a good job as in the case where you use the real receptor mass. And that's opened the possibility to bind the neuromodulator system and to use this binding of empirical functional and neuromodulation in order to explain the emergence of the global dynamics of the brain, and as I said, was my main focus today, to try to provoke in a stimulus-driven way transitions between brain state. And as we see, at least in silico, of course, we are still far from the reality, and our hope is that we can show this also in the reality. And we know in the reality that this happens, because we have seen in the first video that TBS has a tremendous influence at the behavioral level, and of course at the resting state level. We have already seen that to use that in order to design the clinical use of those external stimulation. So thank you very much for your attention.